Good morning, folks.
Good morning. Good morning.
Hello, nice to see everyone. Okay. All right. So hopefully a few more of your classmates will join us here this morning. But in the meantime, we'll get started. Okay. And so welcome back. Hope you're doing well. Hope you had a nice weekend. Get outside if you have the luxury of doing so with the fall weather approaching. So, okay. Thank you, Christian. I see your comment. Thank you, Christian. Okay. So we're going to continue our conversation in chapter 3, right? I have 3 lectures scheduled for chapter 3. Last time we defined some of the major anatomy of a cell. So we're going to move beyond that topic and talk, at least the first topic we're going to talk about today is moving cargo through the plasma membrane. That's a major talking point for us. So we have today on chapter 3. Thursday will also be chapter 3. And then we'll be starting chapter 4 next week. Okay, so things are starting to accelerate. Looking at your syllabus just to stay up with things and make sure that you're not caught off guard by things, right? I recommend you look at your syllabus at least once a week so you're not surprised by anything. We are now 3 weeks away from our first lecture exam, right? Lecture exam 1 is 3 weeks from today. And your very first lab exam of the semester is next week, right? We're going to start that next week. So some very important things starting to come up on our schedule here. Do you have any questions before we continue? Okay, all right, folks. Then let's begin. Okay, I'm going to share my screen with you. All right. So here is our document, our note-taking document. The first thing I want to do is remind you about some of the 3D physiology videos that you have access to in the course, right? Through Wiley Plus, you have videos, 2 of which I'm going to share with you today during the class that are very well done videos. They do a nice job of presenting information visually and explaining some difficult to understand concepts. Don't forget about these things when you're studying during your private study session. Let me just show you what that looks like. Okay, so if I go first here in the web browser to Wiley Plus, okay, so I'm in Wiley Plus, I'm in the lecture portion of Wiley Plus, go to Wiley Course Resources. Underneath the first heading here of course-wide resources, we go down to 3D physiology. I'm going to open that up, and there's 2 videos in here today that we're going to take a look at because they're relevant for our conversation this morning, okay? Okay, so don't forget about those. They can be very helpful. All right, so we're going to talk about moving cargo through the plasma membrane. Okay, this is a major theme for us here. And so I'm going to start with a little bit of information on page 67 of your textbook. Then I'm going to quickly move to the next pages, 68, 69, and the next several pages here. Okay, so when we say, so last time we talked about the anatomy of a cell. We highlighted the plasma membrane made of the phospholipid bilayer. Also, we highlighted the different proteins like integral proteins, peripheral proteins, glycoproteins, glycolipids, and steroid molecules that are embedded throughout the fabric of the anatomy of the plasma membrane, okay? So the plasma membrane, as we said, is the border of the cell, and it regulates what enters the cell and what exits the cell, okay? So let's talk about membrane permeability here just a little bit. Okay, membrane permeability, and I'll make just a couple opening statements here. Okay, so first off, the phospholipid bilayer of the plasma membrane is permeable to certain categories of substances and impermeable to other categories. Don't let the word permeability throw you, right? If it's permeable, it allows material to go through it. If it's impermeable, it does not allow material to go through it. So the phospholipid bilayer of the plasma membrane is generally permeable to nonpolar, uncharged molecules. For example, what would be examples of nonpolar, uncharged molecules? Well, let me give you a couple here. Oxygen. Okay, oxygen. So oxygen, carbon dioxide, and even things like steroids. Okay, steroids, remember, are a category of lipid, and so they are biochemically compatible with the lipid chemistry of the plasma membrane, so they can sneak through pretty easily. Okay, so let me ask a question in the chat window. Here we go. The version of oxygen mentioned in the class notes, is it elemental oxygen or atmospheric oxygen? Do you know what the difference between those two are? Okay, very good. Thanks everybody for contributing. And as you look down there, it seems that we might have a little bit of tug of war, a little bit of disagreement, okay? The answer is the version of oxygen that I'm talking about moving through the plasma membrane is going to be atmospheric oxygen. It's going to be the kind of oxygen we breathe. So what exactly is the difference here, okay? Oxygen comes in many forms. Two forms of relevance are elemental oxygen. And elemental oxygen refers to individual oxygen atoms as they are defined on the periodic table of elements, okay? Okay, that is, with some exceptions, that is generally not the form of oxygen you're taking into your lungs when you inhale, right? Right now, right, you're inhaling, you're exhaling, you're inhaling, you're exhaling, you're inhaling gases of the atmosphere, and we can talk later about all the variety of gases that make up the air in the atmosphere. But generally speaking, you're not breathing in elemental oxygen when you breathe in air, you're breathing in atmospheric oxygen. Atmospheric oxygen is diatomic oxygen. It is two oxygen atoms linked together by a covalent bond. Okay. This is the form of oxygen with two atoms linked by a covalent bond that we inhale from the air around us, okay? So when we're talking about moving oxygen across the plasma membrane, that's the version of oxygen we're talking about. And think about how relevant that is. You inhale, right? You breathe in air into your lungs. Somehow that oxygen in the air has got across the plasma membrane of the cells of your body to get into your body, to get into your bloodstream, to be distributed by your circulation to all the tissues and all the cells of your body, from your brain at the top of your head to your big toe at the bottom of your foot, right? So it's the atmospheric oxygen that's moving across the plasma membrane. Okay, So letter A is our first talking point. The phospholipid bilayer of the plasma membrane is permeable to nonpolar, uncharged molecules. Here's a few examples: oxygen, carbon dioxide, and steroids. Secondly, the phospholipid bilayer of the plasma membrane is impermeable. Okay, it will not allow the passage of, is impermeable to ions and uncharged polar molecules. For example, okay, so examples of ions would be things like sodium ions, fluoride ions. Examples of uncharged polar molecules would be things like glucose, okay? All right, so these sorts of things cannot get through the plasma membrane without the help of some special transporter proteins. I'm not going to list them out in your notes because I have other things to do today, but definitely pay attention to figure 3.3 on page 67, where it talks about the function of membrane proteins, okay? All right, so we've given you some examples of substances that cannot get through the plasma membrane, cannot get through the phospholipid bilayer by themselves, okay? All right, now with letter B, we have to say there are some exceptions to this statement, okay? There are some exceptions, okay? An exception to rule B is the observation that small uncharged polar substances like water, H2O, can get through the membrane. All right. Generally speaking, the smaller a molecule is, the more likely it can pass through the phospholipid bilayer, as long as the molecule does not have a charge, okay? Generally speaking, the smaller a molecule is, the more likely it can pass through the phospholipid bilayer, as long as the molecule does not have some sort of charge to it, positive or negative. Generally speaking, the larger a molecule is, the more difficult it is to get through the phospholipid bilayer. Okay, so this is just some rule of thumb statements to start guiding our conversation today. Also, if you're going to talk about membrane permeability, you have to be aware of the use of gradients. Okay, a gradient is an area where you have a lot of something and another area where you have little of something. It's an imbalance. Okay, so we need to talk a little bit about gradients. Okay, you notice gradients is talked about on page 68 of your textbook. Different types of gradients used by cells to control the motion of cargo. All right, we have the concept of a concentration gradient. Okay, now we talked about concentration previously, so I'm going to assume you remember what that is, right? Concentration is the amount of a solute dissolved in a volume of liquid, okay? So a concentration refers to a difference, and that word is so key here. A gradient refers to a difference, okay? A difference in concentration of a chemical between two locations. Okay, most usually those two locations are inside the cell and outside the cell. Inside the cell and outside the cell. That would be the two locations. All right, so cells will maintain a concentration gradient to regulate the motion of certain kinds of cargo. Other gradients include electrical gradients. Okay, an electrical gradient is a difference in electrical charge between two locations, again, such as the inside and outside of the cell. Okay. So, when we talk about a concentration gradient, an example I could give you here would be a gradient of glucose, where I might have a lot of glucose outside the cell because I just ate a lollipop, because I just ate some food. So I might have a lot of glucose outside the cell, relatively little glucose inside the cell. So the passage of glucose is going to be regulated and constrained by this concentration gradient. An electrical gradient would be something like sodium chloride gradients, where you just ate food that had sodium chloride, table salt in it. Or if you don't like that one, a hydrogen ion gradient, a proton gradient that is very influential in processes where we manufacture ATP. Okay. Sodium chloride. Sodium chloride. Okay. And then, if we're being completely honest here, in some scenarios, there will be electrochemical gradients, where we combine the two ideas of concentration and electrical gradient together at the same time. Okay. Electrochemical gradient. An electrochemical gradient refers to the combined influence of chemical and electrical gradients on the movement of a particular material. Okay, so again, an example here would be potassium ion gradients. All right, folks, do you have any questions right now about permeability or gradients?
Why is a potassium a gradient?
Why is a potassium a gradient? Well, again, it would be due to the potassium you take in from your diet, right? Cells maintain a certain relative concentration of potassium inside the cytoplasm. However, when you eat something and you digest whatever food you're in, it's going to have some potassium in. Okay, so what's a high potassium food? Like a banana. You eat a banana, it's going to have a lot of potassium in. Okay, so as you ingest the potassium, you're going to have an abundance of potassium sitting outside the cells of your body. So the passage of that material will be regulated by this idea of an electrochemical gradient. The chemical part is the actual identity of the atom. In this case, it's a potassium atom. The electrical part is the fact that this atom has a positive charge. Okay, other questions? Okay, so let's start talking about the methods of moving cargo through the membrane. Okay, so the heading here is moving cargo through the plasma membrane. Number 3, methods of moving cargo. Okay, so there's basically 2 major types. There are passive processes. And when I say passive processes, I mean that cells are not consuming any of their own biochemical energy to move the cargo. The cell is not burning any ATP to move the cargo. Cargo is, so let me define it this way. Cargo is being moved without the cell burning its own energy, ATP, to move the cargo. Okay. All right. So, a very rough analogy of this would be kind of like this. If you are at the top of a staircase and you're holding a tennis ball, if you open your hand and drop the ball, won't the ball roll down the staircase all by itself without you doing anything to it? Because gravity will take care of it. That's a passive process because your muscles did not have to push the ball down the stairs. It did not have to push the ball down the stairs. That's a passive process, okay? There are cargoes that can move from a passive process standpoint without cells burning their own biochemical fuel. We'll give you examples of that in just a bit. We also have active processes. Okay, active processes in which the cargo is being moved by the cell burning energy to move the cargo. Okay, so let me just get these analogies in. So the passive process analogy was dropping a ball at the top of a staircase. Ball falls without any muscular energy involved. If we reverse our position, if we are at the bottom of a staircase and holding that ball, and we want the ball to go up to the second floor, aren't we going to have to throw the ball, which is going to imply muscle contraction, which is going to involve the burning of biochemical fuel to contract the muscles to coordinate the motion of the ball up the stairs? That would be an active process. Okay. Okay, there are multiple types of passive processes, multiple types of active processes. Let me list the types out in our notes here. Then we'll start watching some videos, then we'll come back and define them in more specific detail, okay? So different types of passive processes will include the following. Simple diffusion. Simple diffusion, facilitated diffusion, and osmosis. These are all passive processes used by cells to move different kinds of cargo. Active processes, different types. We have active transport. We have endocytosis. We have exocytosis. And we have a weird thing called transcytosis. Okay, so we've listed these things out, and this is meat for our next conversation, probably for at least the next 30 minutes here, okay? All right, so what I want to do is start showing you some of these videos as a way to get you to understand these things. We'll watch some videos, we'll come back and define these things, give you some examples, and hopefully it will start making more sense, okay? Do you have any questions at this moment? Okay. All right, so gang, I'm going to start by showing you the Wiley Plus video on passive transport processes, okay? So let me cue this up. Okay, you have access to this in Wiley Plus. You can always see it again during your private study time. All right, here we go, gang. Enjoy.
The 60 to 100 trillion cells of the human body are surrounded by extracellular fluid containing substances needed by the cell, such as oxygen, ions, nutrient sources of energy, and ingredients for cellular functions. This fluid is also the location for disposal of wastes and carbon dioxide. All of these substances need to be transported across the plasma membrane. Active processes utilize energy to move a substance across the membrane, generally from areas of lower concentration to areas of higher concentration. Passive processes require no energy to accomplish this movement. Gases, ions, and nutrients are transported this way. The structure of the plasma membrane plays a large role in determining the specific transport process utilized by the substance. The plasma membrane is constructed of 2 layers of phospholipid molecules. Each phospholipid has a polar head and 2 nonpolar phospholipid tails. The polar heads face the extracellular fluid, or the cytosol. Their nonpolar lipid tails face each other. This arrangement prohibits electrically charged, or polar, molecules from passing through the lipid bilayer of the plasma membrane. Small, uncharged, and nonpolar substances can pass through freely. The kinetic energy of its atoms or molecules causes a substance to move from an area of high concentration to an area of lower concentration. The difference between the two is referred to as a concentration gradient. If the substance is charged, the distribution of positive and negative charges on opposite sides of the plasma membrane also results in an electrical gradient. The combined influence of differences in electrical charges and in chemical concentration is referred to as an electrochemical gradient. Passive processes occur when a substance travels down its electrochemical gradient, either unassisted or with help of a transport protein. Simple diffusion is a passive process involving unassisted movement across the plasma membrane. Non-polar substances and small uncharged substances, such as oxygen and carbon dioxide, pass through the phospholipid bilayer with little resistance. Ions and polar molecules cannot move through the phospholipid bilayer via simple diffusion. The passive process of facilitated diffusion uses protein transporters to facilitate the movement of substances through the plasma membrane. Channel-mediated facilitated diffusion involves an ion channel, and carrier-mediated facilitated diffusion uses a carrier protein. Channel-mediated facilitated diffusion requires the presence of a tube-shaped ion channel across the plasma membrane. Each channel is selective, allowing only certain ions to pass through. Common ion channels allow passage of sodium, chloride, potassium, or calcium ions. Most ion channels change shape, acting as if they have gates that open and close to allow or restrict ion passage. In another type of ion channel, a leak channel, ions pass slowly but almost continuously through the channel. During this facilitated process, substances diffuse down their electrochemical gradients. In carrier-mediated facilitated diffusion, a carrier protein transports a solute across the plasma membrane. Each carrier protein possesses internal binding sites specific to a particular solute, such as glucose. When those binding sites are occupied, the carrier protein changes shape, opening to the opposite side of the membrane and allowing the solute to pass. A final passive process is osmosis, the movement of solvent rather than solute. In osmosis, a solvent, such as water, moves down its concentration gradient. A higher concentration of solutes on one side of a membrane means a lower relative concentration of the solvent on that side. As the quantity of solutes increases, the osmotic pressure also increases. In general, as solute concentration is increased, so does the tendency for water to move towards those solutes. Osmotic pressure is a measure of the force exerted as water is drawn across the plasma membrane in an attempt to equalize the concentration of solutes.
Okay, gang. So that does a pretty nice job of summarizing these passive process talking points, but you're going to need to spend some private study time making sure you really get what these things are, right? And we're talking about content that was just described from pages 69, 70, 71, 72, 73. All of that content was sort of summarized in a 5-minute video, and you want to spend some significant time thinking about this. Well, let's see if we can give you a little bit of extra help before we move on here. Did you have any questions from watching the video that you'd like to ask? All right, so let me do a couple things here. When we were talking about passive processes, okay, the cargo is being moved without the cell burning its own energy. Okay, so let's start defining these things, these different types in your notes. Simple diffusion is the movement of a solute from an area of high concentration to an area of low concentration. Okay. So think about this. Maybe not everybody, but you know what I'm talking about when I say coloring Easter eggs. Okay. Now there's different ways you can color Easter eggs, right? You get some hard-boiled eggs, and you probably buy an Easter egg coloring kit from the supermarket. A lot of times they have those colored tablets in, right? Like green tablets, pink tablets. You take a tablet, you drop it into a cup or bowl of water, and what happens? Doesn't the tablet dissolve? And all of a sudden, a minute later, the entire volume of water in the bowl is now colored pink or green or whatever color the tablet was. When that tablet dissolves and the pigments spread out through the volume of water, that's simple diffusion. That is simple diffusion, okay? So simple diffusion, you move from an area of high concentration to an area of low concentration, okay? Simple diffusion does not require the presence of a membrane to occur, but is used by cells to move some solutes across the plasma membrane, like O2, diatomic oxygen, and CO2, oxygen and carbon dioxide. Think about that for just a moment. Isn't it a really good thing that the oxygen our bodies need to live will move passively across our cell membranes to allow our cells to live? In other words, isn't it a very good thing that all that oxygen we need, we don't have to burn any energy from our own body to get it into our tissues? That's a really good thing, because if we had to spend energy to move oxygen into our tissues, guess what? We would probably spend all of our life simply breathing and having no other energy left over to do anything else. like take a college course or play softball or dance or work at the supermarket. We wouldn't have any extra energy left over if we had to burn energy simply to get oxygen into our tissues. So it's really good that Mother Nature designed a system that can allow oxygen to enter our body's tissues without burning extra energy. Okay. Facilitated diffusion is the movement. Okay, and I'll leave it to you to pick up the pictures in your book, right? I mean, the principle of diffusion is on figure 3.4, page 69. That would be an example of simple diffusion. But I'm going to go on. Facilitated diffusion is the movement of a solute across the plasma membrane with the assistance of a transport protein from a region of high concentration to a region of low concentration. Okay. Now there's different kinds of facilitated diffusion. For example, figure 3.6 shows you channel-mediated facilitated diffusion. Okay. Channel-mediated facilitated diffusion would be something that you use typically to move ions across the plasma membrane, like potassium or sodium or chloride. There is also the carrier-mediated facilitated diffusion of figure 3.7. And that's typically used to move larger substances that are uncharged but have a little bit of polarity to them. So glucose would be a traditional example here. Okay. So I think there's some words coming up in our conversation that I need to tickle you with and see if you understand what I'm talking about here. So here's a question in the chat window. What is the difference between polar/nonpolar and charged/uncharged? What is the difference? Right? Because up here, when we started talking about moving cargo at the start of our class today, we said that the phospholipid bilayer of the plasma membrane is permeable to nonpolar, uncharged molecules. And then we said it's impermeable to ions and uncharged polar molecules, right? So there's a lot of words floating about right now. Do you understand the difference between polar, nonpolar, and charged, uncharged? Any thoughts about that? Okay. All right. Okay, Phaedra, thank you for just being honest that you do not understand the difference. So we need to talk about this. Okay, we need to talk about this. Okay, so let's go here. Let's go here and just make a point of clarification. Okay. Okay. Think about sodium chloride. Sodium and chloride ions. Okay, in this scenario, the sodium and chloride have charges. They have charges because they have either lost or accepted electrons from each other. So sodium and chloride ions are charged, meaning they are not polar, because they have lost or accepted electrons. Okay. Now let's think about the OH bond of water. Okay, we've talked about this before, right? Consider the OH bond within water molecules. Okay, in this scenario, oxygen is slightly negative, but it is not charged. And hydrogen is slightly positive, but it is not charged because oxygen and hydrogen are in a tug of war for shared electrons. Okay? When we talk about something being polar or non-polar, it's always in the context of a bond where you're trying to share electrons. Okay? Polar, non-polar terminology refers to partial charges when atoms are attempting to share electrons. Charged versus uncharged terminology refers to when atoms are transferring or accepting electrons, but not sharing them. Okay, folks, let me just ask in the chat window. So this goes back to our conversations in chapter 2. Am I making sense? Do you start to see the subtle distinctions I'm trying to make with the language here? Okay. All right, so these subtle things that we're talking about actually make a great deal of difference when we're moving cargo through the plasma membrane. Okay. And honestly, you know, I would be lying if I said we understood everything perfectly, because we don't. And what I can say is if we understood everything perfectly, then we could write a computer program to mathematically model the motion of cargo through the membrane, and it would simulate reality perfectly. We cannot actually do that. Science can't do that yet, which means we do have some gaps in our understanding. We have to admit that. Okay. All right. Okay, so this is simple diffusion, facilitated diffusion. Osmosis. Okay, let me put another space in here so we didn't get too confused. Osmosis is also a passive transport process. Osmosis is a passive transport process. Let me say a couple things. I'm going to define this for you. Even though the video did an okay job of teaching you osmosis, I think there are alternative, better ways to think about it. Let me define it for you this way. Osmosis is the movement of water across a membrane from an area of high free water concentration to an area of low free water concentration. Okay. So key to understanding this is the idea of free water. Okay, free water. So let me ask you in the chat window. What is meant by free water? Okay. All right, folks, what I mean by free water is water that is not interacting with the solute. Water that is not interacting with the solute. If it's not interacting with the solute, it is free. Okay. Free water refers to water that is not interacting with a solute. Okay. Another question in the chat window here. Okay. Why does water have a tendency to interact with most solutes? Why does it have a tendency to interact with most solutes? Can you answer that question? Going once, going twice, sold. Okay, so Russell, to break them down. Russell, that's pragmatically true. It's not quite what I'm fishing for, but that is pragmatically true. Rebecca, doesn't it have something to do with electrons? Well, it does, but again, we're not quite there yet. Okay, Carissa, thank you. Tony, look at Tony's comment. Look at Tony's comment. Water's polar, water's polar. So it has a slightly positively charged end and a slightly negatively charged end. So that means it tends to be attracted to other solutes that have some kind of charged situation, right? And so therefore, water tends to interact with other substances that have slight charges on them, which is why water interacts with most solutes and then tends to break them down, tends to dissolve them. Okay. All right. Okay, folks. So I'm going to give you a YouTube video that I want you to look at in your own study time, okay, for osmosis, because it's going to explain what I've been talking about here if what I'm saying still seems very obtuse to you. Go to YouTube and type in how osmosis works. Okay, I'm not going to show it to you. I don't want to take time to do that. I want you to have some work to do when you get to your study session, but let me just show you what it looks like. There it is, the very first one. It's 1 minute 43 seconds. 1 minute 43 seconds. So watch that video and see if you get what it's trying to tell you. See if you get what it's trying to tell you, okay? All right. Any questions you'd like to ask at this time?
When you were talking about the free water, hold on, let me see, where did it go? Is the aquaporins, is that the free water, the AQPs?
No, it is not, Jay. Thank you for asking. The motion of water across the membrane is more complicated than we are currently covering. And so let me just say it this way. Let me answer your question. The aquaporins are specialized proteins that help water get through the membrane. The truth of the matter is that water is able to get through the membrane by two alternative processes. One using transport through the aquaporin proteins. Jay, when water moves through the aquaporin proteins, that's not osmosis. That's not osmosis. That's a form of facilitated diffusion. When water goes through the phospholipids, not by going through the aquaporins, it just slips through the space between the phospholipids, that's osmosis. Okay.
But in the book, it says like when under the osmosis section, it talks about the aquaporins. That's the only reason why I asked.
Yeah, absolutely. I understand what you're saying. And that is, what page number is that?
71.
Yes, thank you. Yeah, absolutely. Yeah, right. So let's read this. Let me just read from your book. Page 71. I see where you're looking at, and I'm going to read it. During osmosis, water molecules pass through a plasma membrane in two ways. Number one, by moving between neighboring phospholipid molecules in the lipid bilayer via simple diffusion. Okay. And then number two, by moving through aquaporin proteins. Okay. That's how your book's presenting it, and I'm okay with that. I'm just saying that they're, scientists would somewhat disagree about how to characterize these processes, which is why I'm saying when you move water in between the phospholipids, that's osmosis. When you move it through the aquaporin proteins, there are some scientists that say, no, that's not osmosis, that's facilitated transport, okay? So it's tomato, tomato, don't worry about it, who cares? You're not going to get jammed up on an exam about that, but you are right in highlighting that osmosis that water can get through the plasma membrane by two alternative routes. The one we most traditionally think of as osmosis is the one that I'm putting in your notes. Other questions? Okay. All right, gang. So we have covered the passive transport processes. I definitely want to cover the active processes here. I want to get this finished, okay? But I should show you the video on active processes first. So let me do that. Okay. So I'm going to queue up the active transport video here and show this to you. Okay, let's see what this has to teach us. Here we go.
The 60 to 100 trillion cells of the human body are surrounded by extracellular fluid containing substances needed by the cell, such as oxygen, ions, nutrient sources of energy, and ingredients for cellular functions. This fluid is also the location for disposal of wastes and carbon dioxide. All of these substances need to be transported across the cell membrane. Recall that passive processes include diffusion, facilitative diffusion, and osmosis. These processes occur as a result of concentration gradients of the substance, thereby requiring no energy source to move the substance from one side of the cell membrane to another. Active processes utilize energy to move substances across the membrane, generally from areas of lower concentration to areas of higher concentration. Active transport involves protein transporters known as pumps. Other active processes use membranous transport vesicles. There are two types of active transport, primary and secondary. Primary active transport requires energy derived from ATP. An important primary active transport mechanism involves the movement of sodium and potassium across the plasma membrane using a sodium-potassium pump. 3 sodium ions in the cytosol attach to binding sites in the pump. This triggers ATP to bind to the pump and split into ADP and a single phosphate. The released phosphate attaches to the pump, causing it to change shape and open to the opposite side of the membrane. The affinity of the sodium binding sites decreases, causing the release of the sodium ions into the extracellular fluid. The changed shape of the pump now increases the affinity of the potassium binding sites, and 2 potassium ions are drawn into the pump and attached. This action releases the phosphate, and the pump returns to its original shape. The potassium ions are expelled into the cytosol as sodium ions enter the pump, and the process continues. Secondary active transport uses the energy released when one substance moves down its concentration gradient to move another substance, such as glucose, against its electrochemical gradient. The driving force is primarily the movement of sodium ions from areas of high concentration to areas of lower concentration. When two substances are transported in the same direction by secondary active transport, the protein transporter is known as a symporter. Glucose molecules are co-transported with sodium ions from the extracellular fluid into the cell. The occupation of the glucose binding site causes the symporter to change shape, exposing both substances to the cytosol. When the sodium ions move down their electrochemical gradient into the cytosol, glucose is released into the cytosol against its concentration gradient. Antiporters are protein transporters that move solutes in opposite directions. For example, hydrogen ions are transported out of the cell against their concentration gradient as sodium ions move through the same antiporters down their electrochemical gradient into the cell. This movement of hydrogen ions is important in the regulation of blood pH in the kidneys. When larger particles or greater quantities of solutes need to be transported across the plasma membrane, another active process is utilized. Vesicular transport involves the formation of solute-containing spheres known as vesicles. The types of vesicular transport are identified by the direction of the movement. Vesicular transport from the extracellular fluid into the cell is termed endocytosis. Exocytosis refers to vesicular transport out of the cell. All cells secrete materials such as hormones, digestive enzymes, or neurotransmitters via exocytosis. A membrane-enclosed secretory vesicle in the cytosol fuses with the cell's plasma membrane, and its contents are released into the extracellular fluid. Endocytosis occurs in three ways: phagocytosis, receptor-mediated endocytosis, and pinocytosis. Phagocytosis occurs when cells, known as phagocytes, engulf other cells, bacteria, viruses, and even indigestible particles. When the unwanted material binds to receptors on the phagocyte's membrane, it and the cytosol extend to form pseudopods that surround the unwanted cell or particle and eventually engulf it completely in a vesicle called a phagosome, transporting it into the cytosol. There, the phagosome will fuse with a lysosome, with enzymes that attempt to digest the consumed material. Receptor-mediated endocytosis occurs in cells containing specific protein receptors on the surface of their plasma membranes. When molecules such as hormones or antibodies that the cell needs attach to the receptors, it causes the membrane to cave in and surround the molecules. The receptors are returned to the cell's surface and the molecule is released into the cytoplasm. In a third type of endocytosis, known as pinocytosis, the plasma membrane caves in to form a pouch that fills with droplets of extracellular fluid. After it is sealed, the vesicle separates from the membrane and passes into the cell, where lysosomes break down the contained solutes for cellular use. When a vesicle carries substances from one side of the cell to the other, employing both endocytosis and exocytosis, the process is called transcytosis.
Okay, folks. So some really good videos here, and I want to encourage you to use them during your private study time. What I like about them is honestly that they can draw things much better than your old teacher here can do, because I'm not much of an artist. Okay, so please use these to help yourself understand these processes. Did you notice towards the end of the video, it was talking about pinocytosis, and it mentioned that a lysosome was used to open up that vacuole and release the material, right? So you know about lysosomes because we talked about them last time. Okay, so when we're talking about passive processes, let me just get this in here, right? We're typically moving cargo from high to low concentration. That's sort of a big deal here. When we talk about active processes, which is our talking point now, we are moving cargo typically from low to high concentration, okay? And that requires the expenditure of energy to do that. All right, so let's define some of these things. Active transport is the movement of a solute across a membrane with the help of a carrier protein from an area of low concentration to an area of high concentration. Okay, this is like throwing the ball up the staircase because you're going to have to burn energy in the muscle cells to move the ball, okay, from an area of low to high concentration. Okay, I'm going to let you look at these things along the way. We do have various forms of active transport. On page 74 and 75, they define for you primary active transport and secondary active transport. Okay. Primary active transport. Primary active transport involves the breakdown of ATP to change the shape of a carrier protein to allow material to move across the membrane. That's what they're showing you in figure 310. Okay. And secondary active transport. Okay, secondary active transport, you have different gradients that are going in opposite directions. And because you're going in opposite directions, you can move the cargo across the membrane. That would be figure 311. I ask you to spend a little bit of private study time doing these things. The big deal, however, is that you are moving your solute from an area of low to high concentration. Okay, now we have this endocytosis, exocytosis, and transcytosis business, okay? All of these three things here are forms of vesicular transport, okay? So let me put a heading here, vesicle transport, okay? So by vesicle transport, what we're saying is that a vesicle is formed to move the cargo. A vesicle, a structure involving the plasma membrane of the cell, is going to be used to actually move the cargo, okay? A quote unquote bubble or vesicle involving the plasma membrane is used to move the cargo, okay? With endocytosis, there are three major types, okay? We have, as your book had, we have receptor-mediated endocytosis of figure 312. We have phagocytosis, which is figure 313. Now, phagocytosis takes place specifically from the category of cell known as phagocytes. Phagocytes are a type of white blood cell. They're involved in body defense and the actual process of eating a foreign invader like a bacterium or virus is known as phagocytosis. Phagocytosis is a type of endocytosis. Receptor-mediated endocytosis obviously is also a type of endocytosis. And then, we also have pinocytosis here. Okay, let me see here. Pinocytosis. I guess your book doesn't really give you a picture of that so much. Okay. Let's see. Oh, no, it does. Okay. The bulk phase endocytosis, figure 314. Pinocytosis, figure 314. Okay. All right. So in all of these processes, energy of the cell is being consumed to move the cargo. That's the point. That's why these are active processes. These diagrams you see here, such as figure 312, 313, they're not showing you ATP being hydrolyzed. They're not showing you ATP broken down because that's getting, I guess, too much of a chemical detail, but nonetheless, it is being broken down to move the cargo. All right, so for endocytosis, the big deal here is we are bringing cargo into the cell. We're bringing cargo into the cell. For exocytosis, you're moving cargo out of the cell, right? Exocytosis is sort of the opposite of endocytosis. You are pushing material out of the cell. Well, what would you push out of the cell? You might push out waste products of metabolism that, your garbage, your trash of the cell that the cell doesn't want anymore and can't use. It's also possible you might throw out molecules into a lipid bubble through exocytosis that you want to use to spread it through the bloodstream and have that bubble merge with another distant cell that will then take that cargo in and investigate the information in that bubble. Okay. All right. Transcytosis, as the video shared with you, is when you couple endocytosis and exocytosis together. Okay. So you bring something into the cell, you move the vesicle across the interior of the cell, and then you exit the cell on the other side. Okay. This is a coupling of endocytosis and exocytosis. Okay. All right, folks. So I know that's been a bit of a whirlwind. I didn't quite expect to spend that much time on it, but it is an important topic. The topic of how you move cargo through the plasma membrane is deeply important. And I will highlight for you, table 3.1 is actually very nice, is it not? Table 3.1 on page 79 does a good job of summarizing the things that we have just talked about. Okay. Are there questions that you would like to ask? Okay, all right. Okay, well, then let's do this. I'm not sure, we're not going to get this done today, I don't think, but let's see if you really understand this. And by that, I mean, we're going to do a little practical application here. We're going to do a little practical application talking about some of these transport processes. And to do that, let's see, to do that, I am going to make a drawing. And again, I apologize, I'm not much of an artist, but I'm going to do it anyway. Okay. And so if you want to draw along at home, I encourage you to have a nice full sheet of paper. Okay, so let's see if we can make a practical application of cargo transport processes and our understanding of them. Okay, let's talk about glucose regulation in the body in the context of sugar metabolism. And this would certainly relate to disease processes like like diabetes. Okay, so I'm going to draw some things here. And again, I apologize, I'm not the best artist, but we'll do this anyway. Okay, so the first thing is that I'm going to draw a blood vessel. Okay, this is meant to be a blood vessel. Truth be told, it's going to be a capillary, as part of a capillary bed. Okay, and so inside the capillary, you might imagine that we actually have blood, right? We have blood that's moving along, right? It's moving in this general direction, okay? All right. Now let's talk about, in context of sugar metabolism, if this is a capillary blood vessel, then that, capillaries are found embedded in the fabric of organs of the body. Well, probably the most famous organ that we're going to deal with here in terms of sugar regulation is going to be the pancreas. So we're going to have the pancreas here, and I'm just going to suggest that this is the pancreas. Okay, and obviously the pancreas itself is made of cells. So I'm just going to make a large pancreatic cell. Okay, so we have blood vessels, pancreas, and a pancreatic cell. Okay. Now, what I want you to appreciate is that this blood vessel here, right here, this line, this is a blood vessel wall. And what I'm saying is the blood vessel wall is obviously made of cells. The blood vessel wall is made of cells that are linked together like a jigsaw puzzle in a three-dimensional fashion made of cells. Okay. All right. So what we'll say here is that this pancreatic cell, that this is obviously the plasma membrane of this pancreatic cell. Okay, so let's say that we have some glucose, some sugar that's now in the bloodstream, right? Here's some sugar. Because you just ate a meal. Maybe you had pancakes for breakfast or something like that, okay? All right, so here's the deal. What I want to do is have a conversation with you where you tell me the answers to my questions, all right? And so I know we're just about out of time here, but this is something that maybe we could spend 15 minutes finishing in lab today. I think that would be a useful thing for us to do, okay? So here's the deal. We have sugar in our bloodstream because we just had a meal of pancakes or whatever it was you ate. You got some sugar in here. The pancreas is responsible for monitoring blood sugar. Well, it just so happens that this capillary blood vessel is in the overall anatomy of the pancreas. Okay. Again, I suck as an artist, so you kind of have to imagine some of this. Okay. So the sugar has got to leave the blood vessel and get into the space outside the blood vessel but before we get to an actual cell. So let me ask you this in the chat window. This space here that's outside the blood vessel, but out and also outside the pancreas cell, what is it? What is this space that I've just put orange parentheses around? What is this space? Okay, thanks, Lauren. I'm going to go with your answer right away. It is the interstitial space, also known as the extracellular fluid space. Okay, so this is where you're going to find interstitial fluid. Okay, there are very few areas in the body where there's just open, empty space and no liquid present. So if we're not in the blood, we're going to be in the interstitial fluid, right? The interstitial fluid is outside the blood vessels. So the sugar has to go from location A to location B. So here's my question to you in the chat window. Okay, and after we get this answer, we'll pause. What transport process was used to move glucose across the blood vessel wall? Remember, the blood vessel wall is basically made of cells. So I'm saying, how do we get the glucose through the cell from the blood to the interstitial fluid? Okay, thank you, Russell, for answering in the chat window. Okay, Russell, Rebecca, thank you. That is not correct. It's not exocytosis. It's going to be facilitated diffusion. Why is it not exocytosis? Because sugar is a very tiny molecule. It's tiny cargo. We don't need to use exocytosis here because exocytosis is going to be for large cargo, like giant proteins, hormones, that kind of thing. So because glucose is small, we're going to use facilitated diffusion here. Okay, so this is going to be, boy, I just don't have a lot of space here. So from A to B is facilitated diffusion. Okay, again, because the molecule is so small. Glucose is a tiny molecule. Okay, for exocytosis, I would want to have a giant substance. Okay, we need to take a break, folks. Let's take a break. All right, let's take a break. And what we will do is when we come back from break, we will continue this. Tony, you have a question in the chat window. Is this carrier mediated? Yes, it is. This is carrier mediated facilitated diffusion. Good question. So we'll take a break. We will finish our little drawing here to give you a rough understanding of things in 15 minutes or so. Then we'll get into lab content. Folks, it's 9:19. Let's take a 20-minute break. We'll start lab at 9:40. And the Zoom channel will stay open. You don't have to log off, okay? So take a nice break. I see you in 20 minutes, 9:40. Hello, everyone. Let's see if we can refocus our efforts here and get started again. Okay, so please rejoin us if you can. Okay, so before I get started again on my little drawing and sketch, you know, we covered a lot of content over the past hour and 15 minutes, and we're just trying to nuance our understanding of these topics. Do you have any questions you'd like to ask? Okay, then let's continue our conversation. We're back in chapter 3. We were talking about moving cargo through the plasma membrane here. And we're just giving a little example of a practical application. Okay, so I'm going to share my screen. And here we are once again with the blood vessel, the pancreas. You know, we moved some sugar, some glucose from A to B, and we said that would be through facilitated diffusion. And you know, one of the questions that came up in the chat window, well, why is it facilitated diffusion? In this case, it's facilitated diffusion because glucose is a relatively small molecule. So you're not going to use vesicular transport for this. Okay. So can anybody tell me what, well, let me put this into the chat window. Okay. So when I say sugar, there's lots of sugar, but let's be honest, what we're really talking about at this moment is glucose. Okay, so let me ask you this. What is the molecular formula for glucose? What is the molecular formula for glucose? The molecular formula is going to tell you how many atoms are in the molecule. And that's what I want you to appreciate, to show you how big this molecule is. Okay, anybody? Yeah, I know, it's kind of like, you know, you're probably thinking, really, he expected me to know that off the top of my head? Well, okay. C6H12O6, fantastic. Okay, so look in the chat window, folks. There are, in fact, 24 atoms in the glucose molecule. That makes glucose a pretty small molecule. Not as small as water, not as small as oxygen, but certainly much, much smaller than a large protein would be. Okay, so it's facilitated diffusion in this scenario because glucose is small, C6H12O6. There's only 24 atoms in glucose. All right, so let's continue our conversation here. So we have glucose now in the interstitial fluid, and I want to get it from point B to point C. So I want to go from B to C. Okay, so in the chat window, what is the transport process to move glucose from B to C? What's the transport process? Even if you don't know, take a guess. Okay. All right. So Christina is the first one to take a guess, and she's right. Okay. So Amy, you said diffusion, but you need to qualify. Is it simple diffusion, facilitated diffusion? What kind is it? And the answer is simple diffusion, right? Because from B to C, all we're doing is moving from one location of the river to the other location of the river. There's no membrane involved here. So that's going to be simple diffusion. So from B to C, it's simple diffusion. Okay, folks, does it make sense to you why A to B is facilitated diffusion, but B to C is simple diffusion? Do you understand why that is true? Right? From A to B, you got to cross the membrane, right? From A to B, you got to cross the membrane. From B to C, you do not have to cross the membrane. Okay. Please let me know if you have questions. All right, so let's continue our journey. And let's say we're going to move the glucose into a pancreatic cell. So we're going to go from C to D. And I'm sorry, I don't have a lot of space here, so I'm just going to use what I have. To go from C to D. Okay, so in the chat window, C to D, what form of transport is involved? Okay, so Lauren, it would not be endocytosis. Okay, glucose is too small here. We're not going to use it that way. Okay, it is once again going to be a version of facilitated diffusion. Okay, it's going to be facilitated diffusion. Okay. And you tell me in the chat window, is it going to be channel mediated or carrier mediated? Okay, so yes, it's going to be carrier. Channel is used for small ions like hydrogen ions, sodium ions, potassium ions, chloride ions. It's going to be carrier mediated. Okay, good. So this is going to be carrier mediated facilitated diffusion, right? A to B was also carrier mediated. I just didn't write it down. All right. So at this point, we got to talk. At this point, I'm going to mention a few things that we won't go into detail yet because we just don't have time today to do that. But I will be taking the time next class to go into great detail about what I'm about to say. So remember, the pancreas as an organ, one of its major jobs is to monitor the blood sugar level. So if we have sugar leaving the bloodstream and entering the pancreatic tissue, the pancreatic cells, the presence of the glucose is going to tell the pancreas, oh, there might be an elevated sugar level in the bloodstream at this time, right? Because I'm getting sugar entering my cell. So the presence of the sugar, the presence of the glucose in the pancreatic cell will trigger an awareness of the pancreas that in fact blood sugar was elevated. Why was it elevated? Because you just ate a meal of pancakes. Okay, so long story short, and with taking a little bit of artistic license at this moment to skip over detail, which I will cover later. The pancreatic cell is going to be aware that glucose levels are up in the bloodstream. Okay, so glucose, sorry, my pen is not working so good here. Let me do that again. Try again. Glucose is up. So the pancreas becomes aware that glucose is elevated. Now that's going to lead to a lot of responses of the pancreas, including releasing insulin that's available into the bloodstream and also stimulating the production of insulin. Okay, so I'm just going to highlight a couple statements here. So glucose is up, pancreas cell makes insulin. Now, the process of making insulin is something we're going to talk about next class. I don't want to take time now because it's really involved. So the pancreas cell is going to make insulin, and I'll explain how that is done next class. Okay. So the pancreas cell makes the insulin, and let me tell you something in the chat window here. I would ask you what the molecular formula of insulin is, but there's no way you're going to know off the top of your head, and I'm also sure your textbook doesn't have it. So I'm going to tell you the molecular formula for insulin in the chat window. Here it is. Hold on a second. Quite large. Here it comes. Here's the molecular formula for insulin. 257 carbons, 383 hydrogens, 65 nitrogens, 77 oxygens, and 6 sulfur atoms. So I don't even have a tally on that. Let me get my handy calculator out here. Let's add that up. 257 plus 383 plus 65 plus 77 plus 6. There are 788 atoms in the insulin protein versus how many atoms did we say for glucose? 24. So my point in telling you this is that insulin is tremendously larger than glucose. If I take the number of atoms in insulin and simply divide it by the number of atoms in glucose, insulin by one metric is at least almost 33 times larger. Okay? So insulin is large cargo. I'm telling you that. Hint, 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 hint. Insulin is large cargo. Okay. So what I'm saying to you is through processes that, again, we're not going into great detail with, the cell is going to end up with some insulin. So I'm going to put the insulin here as a triangle. The insulin is made by the cell, and the insulin has to go from location E to location F. So please tell me now in the chat window what the transport process would be to go from E to F for a large molecule like insulin. What would the transport process be? Okay, I'm going to run with the first answers. Tony and Sierra, absolutely. It's exo because it's going out. It's exocytosis. That's absolutely right. Exocytosis is going to be used in the primary region here is insulin is such a large piece of cargo that it has to be thrown out of the cell. Okay. So we go from E to F, and insulin is going to be moved by exocytosis. Okay. Now, so insulin at this moment at location F is in the interstitial fluid, correct? So the insulin, let's see what happens when the insulin goes from location F to location G. Okay, so from F to G, what would the transport process be? From F to G, what would the transport process be? Okay, so thanks to everybody that's thinking about this and trying to work it through in your brain. That's fantastic. Really appreciate that. And the answer, of course, is simple diffusion, right? Simple diffusion. Okay, because we now have the insulin in the interstitial fluid. We're not moving through any membranes, so we're simply going from one location of the river to another location of the river. There's no membrane. So again, that's going to be simple diffusion. So F to G. I'm running out of space here, but isn't it kind of interesting that for insulin, which is a large protein, to go through the plasma membrane from E to F, we have to use exocytosis. But then that same large protein simply floating in the river can go to two different sides of the river, F to G, by simple diffusion. Okay, same cargo, but because of its location and the context of its location is moved by different processes. Okay, so questions so far? Questions so far? Okay, now here's what I want to do with you. One last challenge. So we have insulin approaching the blood vessel wall, and the blood vessel wall is drawn with a black line, and I'm telling you that the blood vessel wall is made of cells. So this area right here that I'm now putting into a blue rectangle, I need to magnify it and draw it in more detail for you. So see below. Okay. So folks, I apologize. I don't have enough screen space here. So in the count of 3, I'm going to go low and redraw part of the blood vessel. Okay, I'm going to go low and redraw part of the blood vessel. Here we go. I'm going to go down. Okay, so this is, okay, so here we're going to redraw the blood vessel. And so the blood vessel is going to look like this. So here's the top of the blood vessel as you saw it. But what I really want you to appreciate is that blood vessel wall is actually made of cells, right? Cells. So here's the bottom of the blood vessel wall enlarged by cells. Okay, and so we have insulin at location G. I'll go back to my orange. So we have insulin at location G, right? Insulin is here. And the question is, how do I get insulin from location G into the blood vessel cell, through the blood vessel cell, and out of the blood vessel cell so that insulin is now sitting in the bloodstream? How am I going to do that? And hopefully my language that I'm using tells you what the answer really is. So how do I take insulin from G to H? It is not endocytosis. It's not endocytosis. It's not active transport. Sierra, okay, good. Here we are. Take a look at Sierra's answer. Sierra's answer is right. It's going to be transcytosis, right? Transcytosis. So G to H. Remember from our definition above that transcytosis is what takes place when you couple endo and exocytosis. So for some cargo like insulin, it has to access the bloodstream. It has to enter a cell that's part of the blood vessel wall. It has to go through the interior of that cell, and it has to exit that cell to get to the actual bloodstream. So you enter by endocytosis. We use additional vesicular transport processes along the cytoskeleton inside the cell, and then we export that material by exocytosis. So when we do that all together as one uniform process, endo coupled to exo, enter the cell, go across the interior of the cell, exit the cell, that's considered transcytosis. So if you answered endocytosis, you're not wrong. It's just that you only gave us half the answer, right? You got to couple everything together. When you see that happening, when you couple the endo and exo together, that's going to be considered transcytosis. Okay. All right. So let me just ask in the chat window. Right, this is a lot of new content, a lot of things you need to process and put in an organized way in your brain so you understand. It's part of the learning process, right? It's part of the studying process. It's part of you spending study time to think about these ideas. Okay, so again, glucose is small, which explains why for many cases you simply use facilitated diffusion, but insulin's much larger, which is why you got to start engaging the vesicular transport processes of endo and exocytosis. Okay. So, it's one thing to write definitions down during a private study time. Just, you can memorize the definition, but ask yourself, are you really getting to a true understanding if you can't start to apply it? That's what we're trying to push you forward. Okay. Anything for the good of the order here before I stop this topic? Okay. All right, then I'm going to let it rest. So next class, next lecture on Thursday, I won't really talk about this anymore. What I am going to talk about for the third and final lecture of chapter 3, I'm going to talk to you in great detail about the process of protein synthesis starting on page 93 and going forward to the next several pages. Okay, so that's where we're headed with lecture in chapter 3. Okay, gang. Thanks for your attention. I'm going to stop sharing my screen at this moment. And I'm also going to stop recording because that completes the lecture portion